Hello, everyone. Welcome. Looks like people are coming in from backstage. Thank you all for being here today. Typically, I let a minute or two go by just to let people come in and join, but I'm going to go ahead and get started today because we have a lot to go through and a lot of great speakers, and I want everyone to learn as much as possible. So I'm going to start. My name is Ellen Hostetler, and I'm the Chief Science Officer for the John Ritter Foundation for Aortic Health. We are so happy to have you all with us for kind of a new new topic for us to discuss in these webinars that we do. I'm just going to go ahead and jump in with some housekeeping too. There is a Q&A bubble at the bottom of your screen. If you have questions for the presenters, please enter them there. You can even choose to do it anonymously if you don't want your name attached to it. There is also a chat that you can talk to your fellow attendees, but if you have questions, the Q&A is the best place for those so that we don't miss them. Last, just, be kind and respectful in the chat, of course. And if you have any questions or concerns, you can always email me, this is recorded, so that will be available later, and we'll send that out to everyone who's on this registration list, and it will also be on our website and our YouTube channel. So with that, I am going to get started with our first presenters today. You can see them all on screen here. We have a great group of people to talk to you about very cool research that they're doing, and how what the work they're doing is really going to be a huge benefit to the patients and families and everyone else in our community. But we're going to start off our first presentation with, and I'm going to pull up my little intro to make sure I get my details right here, Dr. Pedroza and Dr. Dalal, and they are both residents in the Integrated Cardiothoracic Surgery program at Stanford. And one of them just finished, and the other one is currently doing a NIH-funded research fellowship in thoracic aortic disease research in Dr. Michael Fishbein's laboratory. And Dr. Pedroza's research focuses on dynamic gene expression changes driving aneurysm development using genetic mouse models and human surgical tissues. And Dr. Dalal's research focuses on molecular mechanisms of aneurysm formation in genetic aortic disease. So some of those words were a little complicated, so hopefully their presentations are going to explain exactly what that means in more detail to us all. And I will turn over the screen to them, so feel free to share your presentations and go ahead and get started.
Thanks for the introduction, Ellen. We're both, I think, really excited to be here. I think this is a really unique opportunity for us to interact with the patients we hope to serve. And as you mentioned, Alex and I are both trainees, both in science, but also in cardiac surgery. And so these are obviously topics and diseases that we're very interested in, both in the clinical realm and also from a research standpoint. So this is a really awesome experience for us to be able to share some of that with you guys. And I will try to keep this as topical as we can, and just try to make it so you guys can get a sense of what we're up to and why we think this stuff is important. So the title of our talk is Brick by Brick, and we're going to talk about how we dismantle the aorta to learn something about the way aneurysms progress. Just as a brief overview, this might be too simple for most of you, but when we think about the aorta, in its most simple sense, it is a tube, and I think Dr. Wagenseil is going to give us a lot of information more about the extracellular matrix and elastin structure. But I'll just start by saying that we think about the aorta in layers, and these are called what are called tunicas. And it has basically three major layers: the intima, which is the innermost; the media, which is in the middle; and the adventitia, which is on the outside. And we know from decades of research that the most visible changes that occur in the aorta during the development of aneurysm, and certainly during dissection, occur in this media layer. But it's our belief, and I think a lot of people's belief, that every part of the aorta has a role in this, and every cell type has a role in this. And that's why we sort of like to think about this within its component parts. And so in terms of the cell types that are involved here, on the intima layer, we have what's called endothelial cells. And we're not going to talk about much of that today, but we focus our work mostly historically on smooth muscle cells, which are in that media layer. And then on the outer layer, the adventitia cells, which consist of mostly fibroblasts, but also some smaller vessels, some nervous tissue, and some inflammatory cells. And Alex is going to talk a little bit about that layer. But suffice it to say that it's more complicated than simply a hollow cylinder that transmits blood. And I think any patient who's had an aortic problem can understand that this is a dynamic organ that has its own series of problems and challenges to deal with. Sorry, we're multitasking here with the controls. And this is just a picture of some of those aortas to give you a sense of what we're looking at. And so on the left side is what's called a trichrome stain, and that gives us a look at the collagen, which is one of the main proteins within the aorta. And you can see that in blue there, and the red is actually the cells. And then on the right side is what's called an EVG or elastin von Giessen stain. It actually looks at the elastin layers. And elastin gives the aorta its elastic or stretchy properties, and also its resistance to rupture with pressures. And you can see those black squiggly lines, which represent the layers of elastin within the aorta. Our work principally is studied in patients with connected or hereditary aneurysm and dissection syndromes, chiefly Marfan syndrome and the growing list of different varieties of Loeys-Dietz syndrome with mutations in the genes you see listed here. But we think that some of the principles that we're studying and the behaviors of these cells will ultimately apply to patients who have an aortic problem stemming from a variety of problems or a variety of causes, whether that's hereditary or sporadic aneurysms and dissections. And so we study these diseases in part because they're very predictable in terms of the way that they present, particularly in animal models. But I just want to make the point that everything we talk about, we think is relevant to the sort of whole world of aortic diseases. As Ellen mentioned, there's a lot of complexity in words that we use when we talk about these things, but I like to use metaphors to try to break this down. This is useful even for us and for our students in the lab. And this concept, what's called the central dogma of biology. And basically the concept here is that you have a forward flow of information from DNA to RNA to protein. And the metaphor I like to use for this is sort of like the DNA being like a textbook or a book that has the instruction manual for how to put something together. Let's say how to make a bench. And so that's one copy of information that has to be stored and taken care of. But you can make photocopies of the individual pages, and that's what I would call an RNA. Basically, is a copy of that information that's transmitted and is usable. So if I make 50 copies of RNA, now I've amplified my signal, and I have, instead of one copy, I have 50. And it turns out that those photocopies can then be used by individuals to actually build that piece of equipment. And so that's what the protein represents, is actually not just information, but actually a structural piece of molecular machinery. And so this is the way I like to think about it. And our research focuses primarily on the intermediate step, that RNA, because it tells you something about how the cells are thinking, which pages they've chosen to make copies of, which pages they've chosen not to make copies of. And it gives you a hint about the way that they might be constructing their environment, making those proteins, building those things from the message, and using that to perform actual cellular functions. So that's kind of the simplest way I can think to explain that. In terms of why we think, so our most of our recent research has centered on the use of what we call single cell analysis. And the reason, to give you an example of why we think this is important, I've used a lot of metaphors for this in the past. This is actually Alex's metaphor, which I think is the best one I've heard. It would be like, basically, if you were at the store and you saw a bag of M&Ms and you wanted to know, okay, looking at it from the outside, like what are M&Ms? Like, what if I had to describe them, if I had to give you information about them, what would I want to do, right? And so what you wouldn't want to do is put into a furnace and melt it down, because you would think, okay, I have a puddle of brown chocolate, and it's just one big mass of chocolate, and this is what it looks like. But in reality, we all know that if you cracked open that bag of M&Ms and you started to classify the different items within the bag, you'd see that there are different types, there's different colors, and that this is the sort of information we hope to glean by taking the aorta apart and looking at it as component parts. And I'll show you what that means to us in just a second. I should say that, historically, the way that we would study RNA or protein analysis would be exactly the first version, the melting analogy. And so we would take a piece of tissue, we'd grind it up in some kind of buffer, and we would extract either RNA or DNA or protein. And we'd say, okay, what's in my sample? And we'd compare a disease sample to a patient who doesn't have a disease, or animals with Marfan syndrome versus ones without. And that gave us a lot of useful information, but it totally clouds the picture of what's going on at a smaller level. And so a few years ago, we undertook this new technology to basically take the aorta and digest it. So we use what are called enzymes. So we kind of melt the tissue apart and let the cells kind of leak out into a suspension of single cells. We collect those cells and we study them individually by looking at all the RNA in each cell. We say, for each cell we can find in our sample, tell me every gene that is expressing all the RNA. And we look at everything and we sort of classify these cells. And we do some bioinformatic analysis to get a sense of what's going on. And so our goals have always been, number one, to harness the resolution of these single cell analyses to determine which cells are actually involved in the breakdown of the aorta. And then to use that information to try to find out what are the signals that are driving these processes? What are the higher level manipulators that we can potentially intervene upon when we create therapies? And finally, are there pathways that we can use from these models of disease that will be useful to us in patients with aneurysm from a variety of causes? And so that's always been our sort of three main goals from these studies. I'll go quickly through one of the studies that we published recently using the Marfan mouse model. And on the top left, you can see an echocardiogram, which is just looking at the size of the aorta. And you can see that the Marfan, which is labeled FBN1 mouse, has a much larger aorta, and this is an aortic aneurysm. We don't see dissections in these mice, but we definitely see large aneurysms. And we have found that to be a useful model to study this process. And we did exactly what I just described. We took these aortas, we digested them up into single cells, and we analyzed them. And these plots on the bottom are a little bit difficult to understand, but the bottom line is we've taken each individual cell, we've tried to express it by putting it into two dimensions. And this is basically trying to take every person you've ever met and describing them with two words. And if you plot those onto a single graph, you can see that the different colors represent different types of cells. And one thing that jumped out at us immediately is that in the Marfan mice, there's this cluster of blue cells, which almost barely exists, almost doesn't exist at all in the control animals. And we learned that these are actually a type of smooth muscle cells, which are those cells I mentioned before in the middle layer of the aorta, and that they have a unique, we call phenotype or behavior. And I'll talk about that in just a second. So the first thing we asked ourselves was, well, what are these cells doing? What makes them different from normal smooth muscle cells? And there's hundreds of genes. If we looked at every single gene, we would just be still looking at this list. We tried to classify them into processes. And it turns out that these genes are all involved in a lot of things that we know to be important in aortic aneurysm. Things like cell migration, proliferation, growth, the assembly and organization of those proteins between the cells, the extracellular matrix, adhesion, so how the cells adhere to their environment. So we know these cells are important because they're doing all the things we know are important in this disease process. And so that's why we sort of thought it was worthwhile to chase these cells down a little bit more. We also wanted to prove that the cells actually reside within the tunica media layer. And so we found some markers, and here you can see in blue our expression of those genes that we identified as being markers for those cells. And we can see conveniently they're blue like in the slide, but these cells are not actually blue. They're being stained with a chemical that actually makes the cells expressing certain genes show up in blue. And we were able to prove that these cells are exactly where we thought they might be, within the wall of the aorta in the layer that's most degenerated. The other thing that we find useful about this is that we've learned that cells don't just go from 0 to 60. This identity switch is not an all or none principle. These cells actually undergo a process that's very continuous. So they don't go from A to B, they go from A to 8.1 to 8.2 to 8.3, all the way up to 8 million up to B. And so we can take advantage of that in our data set because we have thousands of these cells. And by looking at every single one of them, we can capture cells that are at many different points across this spectrum. And we can learn what these cells do in terms of which cells are turned off first, which ones are turned on first, which ones are markers of a very late or very abnormal cell, and what does a normal cell look like. And so I won't go through the details too much, but these plots on the right-hand side just show us some of the different ways that cells can be turned on or turned off. And so these top layer, you can see the cells are expressed very highly at the beginning, and these genes sort of turn down as the cells progress towards a more dysregulated cell type. And the inverse is true for these other genes, which are expressed at sort of different characteristics. You can see these are markers of late change. And so that's information that's useful to us to understand what are the things that are happening earlier in time that might be initiating events versus things that are more related to late degeneration and end stage of the disease. And, oh, sorry, go ahead. I'll turn it over to Alex and I'll talk about another model, and then we'll wrap up in a couple minutes here. I think we're trying not to go over, so thanks.
So to build on what AJ just said, we're looking at another disease model called Loeys-Dietz. We have a mouse model too, and we can see that this mouse, just like the Marfan mouse, develops an aortic root aneurysm. And so we decided to do a similar study and look at the cells from a single cell level. And we expected to see a similar thing. So here on the left, we can see all of our different types of cells, smooth muscle cells occupying the space down at the bottom. But when we compared the TGF-beta receptor 2 mice to the wild type mice, we noted that there was no modulated smooth muscle cell cluster that was specific to the TGF-beta receptor 2 mice. But instead, we started to see that there were a couple other clusters that emerged with differences, being the fibroblasts and the macrophages. So we looked at the fibroblasts a little bit more closely. These are the cells that reside within the adventitia, the most outer part of the aortic layer. And we saw that many genes were upregulated, specifically the collagens and many inflammatory markers that recruit macrophages to the aorta. So we started thinking that this could be a different disease process, not quite identical to Marfan. And so when we went and looked at the tissue again, we saw that these sort of modulated fibroblasts resided in the adventitia just where we expected them to be, and not within the medial layer like we saw in Marfan syndrome. So then to sort of tie this together with our human patients, we have also had multiple patients who have had operations for aneurysms who are multiple types of LDS. We collected their tissue and we collected donor tissue as well. We performed single cell RNA sequencing, and from the fibroblast subset again, we can see this emerging cluster of cells specific to just the LDS population. So we're starting to gain some insight into what might be, that these cells might be important for aneurysm formation in LDS, and how it might be different from some of the other aortopathies that we study. I know we're exceeding our time limit here, so I'll just quickly go through some of the other work that we're working on right now. So AJ had talked about that central dogma. We've been studying the RNA, but we want to start seeing a little bit upstream. So we're studying transcription factors, which bind to the DNA and are sort of a master regulator of multiple gene sets. And they act as like a gas pedal or a brake to increase or decrease some of these processes. So in some of our early work, we showed in Marfan syndrome that a transcription factor called TWIST1 was elevated in the tissue at the protein level and was also elevated at the DNA level. Trying to understand how this affects the Marfan disease process, we overexpressed and blocked TWIST1, and we could see that it regulated certain genes that were specific to aneurysm formation, here specifically collagen 1 and lumacan. So the idea is that we can now start to regulate multiple processes, multiple genes, instead of just one at a time, to hopefully affect the natural progression of aneurysm formation and identify targets for therapeutics. So with that, we'll turn it back over to the rest of the panelists. Thanks for hosting us.
Thank you both so much. I loved the M&M metaphor, so kudos on that. That was really helpful. And I think I had a few questions come in, but if you all are okay with it, I'll wait until the very end and we can do them with all the presenters at once, if that works for you.
Yeah, I will probably have to leave in a few moments to run to the OR, but Alex will be here and he can take any of the questions.
Yeah, well, we are very thankful for that, and that you're working that as I can speak for everyone. So sounds good. Thank you both for being here. I'm going to move to our next presenter, Dr. Kwartler, and there she is. Great. And so I have known her for many years because we used to both work in the same place. So it's very exciting for me to have her on today. And Dr. Kwartler is an assistant professor of medical genetics at the McGovern Medical School at UT Health Houston. And she works alongside Dr. Milowitz, who many people in our community and attending this webinar today know because she's often on the webinars herself. And Dr. Kwartler's work uses mouse models and induced stem cell models to investigate genetically triggered disease. So I will hand it over to her to share her slides and start a presentation. Thank you for being here.
Hi, everyone. Thank you so much for this opportunity to talk to you today. As Ellen mentioned, I work closely with Diana Milowitz, who I think you are all familiar with. And I'm going to be talking today about a project that I'm doing with Dr. Milowitz on the mechanisms of thoracic aortic dissections. I also started with a slide. I'm sorry, I don't know why it's not advancing. I also started with a slide on the structure of the aorta. It has a few different details from what AJ and Alex showed, so I'm going to still go through it. As they mentioned, there are 3 distinct layers. I focus primarily on smooth muscle cells, which are in this middle layer. If we look here in the center of the screen, there's a piece of tissue from a normal human aorta. And you can see with this particular stain, there's interspersed red and black. And the black are the elastin fibers and the red are the smooth muscle cells. Looking next at this graphic, you can see what this looks like more close up. The elastic fibers have the smooth muscle cells aligned between them, and there are links between the fibers and the cells, which allows the whole tissue to contract together. Within the smooth muscle cells themselves, there's this lattice-like structure of contractile filaments, which you can see on the bottom graphic right here. And again, those are connected up to the elastic fibers, allowing the whole tissue to contract. There are two types of contractile filaments that I'll talk about today in the smooth muscle cells. One are filaments made up of actin, which are also sometimes called thin filaments, and the other are filaments made up of myosin, which are called thick filaments. I hope that you can see my hands, but what happens is that the myosin actually interacts with the actin and pushes it, and this allows the filaments to slide against each other and shorten or contract the cell. We come at looking at this disease from the perspective of studying genetics, and there are a couple of reasons why we think this is an important and useful way to think about studying disease. The first is, of course, that if we can identify people who are at risk of aortic disease based on their genetic variants, we can help improve outcomes for those people as individuals. But the second and perhaps more globally important reason to do it this way is that we can start to understand pathways leading to aortic disease. We can use the genetic variant as a starting point to try to figure out what the molecular changes are that are leading to disease. And we think that these same molecular mechanisms represented here by the question marks will be responsible for disease in patients who don't necessarily have an identified genetic trigger. Dr. Milowitz has found pathogenic variants in the genes encoding the actin and myosin that I talked about on the last slide that lead to aortic disease. And today I'm going to be talking about one variant specifically in the gene encoding myosin. Myosin is a really big protein. It has multiple what we call domains, which are different parts of the protein that have distinct functions. Most of the pathogenic variants that we find running in families to cause aortic disease are deletions in what is called the tail domain of the myosin. The tail domain is the part of the protein which interacts with other myosins to form those filaments. But we've also found variants of unknown significance in the myosin head domain that appear more frequently in patients with aortic disease compared with the general population. And the head domain is the part of myosin that interacts with actin and does this movement. So it's also sometimes called the motor domain because it has this motor function. Variants of unknown significance, or VUSs, as you can tell from the name, are genetic changes that we can't tell just from the genetics alone whether they cause disease or not. So we wanted to ask whether this particular VUS that we're thinking about causes disease, and to do that we made a mouse model. This detail is perhaps not important to understanding this story, but the particular amino acid change is arginine 247 to cysteine. So where you see R247C, that's me talking about the VUS. And WT is wild type mice, or mice that don't have the VUS. So the first thing we thought was, if we have a mutation in myosin, it probably affects the contractile function that I talked about at the beginning. The first thing we did over here on the left is look at myosin just by itself in a dish. This is not inside a cell or inside a tissue, but it's just myosin on its own. And we looked at how effectively it could do this motor function. And we found that the VUS significantly decreased that function. Then on the right, in our mice that have the VUS in their gene, we look to see if the whole aortic tissue has decreased contractile ability, and it does. But what we didn't find was any sign of disease. So when we looked at the mice with the VUS, we didn't see any aneurysm, we didn't see a dissection. And if we look at just the structure of the aorta by the pathology over here, you can see at this level, at least, that there are no changes. This was not a big surprise because, again, this isn't a pathogenic variant. We don't see it running in families. So we thought perhaps as a VUS that it's more of a contributing variant. And we asked if we could push these mice to induce disease in them. And the most common risk factor for aortic disease in patients is hypertension. So we asked, what happens to this mouse model if we increase the blood pressure? We did this by feeding the mice a high salt diet and also giving them a drug. And mice have a similar normal blood pressure to humans. the top number should be 120 and the bottom number should be 180. This graph is just representing the top number. So 120 at the baseline, and then when we start this high salt diet plus the drug, we quickly see the top number increase to 160 or even 180, which is definitely hypertensive. Within the first 2 weeks, we started to see about 20% of the mice carrying the VUS died suddenly. In comparison, only one mouse that was wild type died. And I'm not going to show you the gory pictures, but we went through each mouse that died and looked for the cause of death, and we're able to show aortic root dissections were the cause of death of the mice carrying the VUS. However, there was no aneurysm formation preceding the dissection. And this is really important for a few reasons, going back to our reasons to use genetics as a tool. First, we can tell patients who have this particular VUS that it's really important for them to control their blood pressure. That's an immediate impact for those specific patients. But secondly, we now have a mouse model where dissections occur without aneurysms. And because we're most interested in preventing dissections as the catastrophic bad outcome for any patient, we can use this model potentially to start finding those molecular changes that specifically lead to dissections without confounding factors that are a part of aneurysm formation. So we wanted to really get at what those mechanisms are in this mouse model. And the first thing that we did was we used a special kind of microscopy to look deeper at the aortic structure. I showed you a picture much like the one on the top here earlier in the presentation, where the elastic fibers are between the smooth muscle cells, and they're connected with the fibers reaching out via these microfibrils to touch the smooth muscle cells here. And this is the wild type mouse without the VUS. It looks very similar to the picture. So here's the elastic fiber and the smooth muscle cell, and then the microfibril represented with this arrow connecting them. But in the mice containing the VUS, we see a different pattern. Here's the elastic fiber and then the smooth muscle cell. And what we see is the cells are reaching out to the fibers rather than the other way around. When you add hypertension in both systems, you start to see this increase in buildup of stuff that's in between the fibers and the smooth muscle cells. And that just adds increased strain on those connections. The sites where the cells connect to the elastin are called focal adhesions. And we did the dramatically chocolate soup version of looking at what genes are expressed in the aortas of mice that have both hypertension and the VUS. And what we found was that the most upregulated pathway was focal adhesions. This is not surprising given the structural changes that I showed you on the last slide. One of the key proteins inside of focal adhesions is called focal adhesion kinase, which I've abbreviated here as FAK. And it gets activated by something called phosphorylation. So PFAK means activated FAK or activated focal adhesions. This data that I'm showing you on this slide is done by a technique called a Western blot. It is frankly a pretty annoying technique, but it's the best way we have to look at whether a specific protein, how abundant a specific protein is in our samples. And you can tell the size plus the intensity of these bands indicates the amount of the protein of interest. So these are the mice with the VUS at baseline here. And you can see there's more activated focal adhesions, more PFAK in the mice with the VUS compared with the wild type mice. Over here, I'm looking at just the mice with the VUS. This is at baseline and then over the course of the 2 weeks critical window of hypertension. And we see that the PFAK or activated focal adhesions are increasing over that time frame. So what I've shown you so far is that our VUS of interest is causing aberrant aortic structure. And when we add hypertension, it increases the strain on the focal adhesions and activates focal adhesion signaling. I'm going to go through a little bit of what I'm talking about here in gray. Now, I'm going to take a step back and try to explain the context before I show you a little bit of the data. I don't want you to get too worried or bogged down about the specific details. I'm going to try to explain the pathway first. So the word energy appears in the last slide, and your cells need energy to function. And the currency of energy that your cells use is called ATP. I specifically talked a few times about myosin moving actin like this, and myosin needs ATP to be able to make that motion. And what happens when focal adhesions get activated is that there's a sensor on the edge of the cell, and they're saying to the rest of the cell, hey, we're under a lot of strain. We need to do something about this. And what it's telling the cell to do is have the myosin go faster, which means more ATP is going to be needed. Where does that ATP come from? How does it get made? You may remember details from high school biology, you may not, but the brief answer is that ATP is made in the mitochondria. Again, don't worry about the details, but one of the byproducts of this reaction where ATP is made is called reactive oxygen species. And in particular, if ATP production increases too much too quickly, you get more reactive oxygen species. And reactive oxygen species can cause damage to other proteins in the cell. And if that damage starts to accumulate, then the cells end up dying. And we think that's what's happening in our mouse model with the VUS. I'm just going to show you a little bit of the data now to back up that hypothesis. Here I'm using cells in culture with the VUS. And that means that we're not in the context of the whole aorta. There are no elastic fibers or anything. So we had to come up with another way to activate focal adhesions. And what we did was we added this protein called fibronectin to the cells. And we can see using our PFAC marker that immediately we start to get activation of the focal adhesions. Then I want to draw your attention down here. PRLC is an activation of the myosin that we are using as a proxy for the amount of energy demand in the cell. So you see on the same short time scale that the energy demand goes up dramatically when you add fibronectin. In the last lane over here, I'm using an inhibitor of the focal adhesions. So you can see that I have effectively blocked both the activation of the focal adhesions and that increased energy demand, which means that focal adhesions activation is leading to the energy demand, and that allows us to figure out what direction the arrow is going in our pathway. The next step in the pathway that I talked about was accumulation of reactive oxygen species. And to assess this, we put a dye on our cells that becomes fluorescent when it reacts with reactive oxygen species, and then we measure the amount of fluorescence. And I know this is a little complicated just looking at graphs, but the line is in the same place in both of these graphs. And you can see that in the wild type cells, the curve is centered around the line. But in the cells with the VUS, the curve is shifted to the right along the x-axis, which means that there's more fluorescence being red, and that means there's more accumulation of reactive oxygen species with the VUS. The last part of the pathway that I talked about was increased cell death. We're using here a marker of one particular gene that we know from the literature, from prior research done by us and others, that this marker increases when the cells are dying. So we use it again kind of as a proxy for looking at what we think is happening. The white bars here are the wild type cells, and these are the cells with the VUS in black. You can see already at baseline that the VUS containing cells have more cell death marker. And if you add the fibronectin to activate the focal adhesions, cell death marker increases even more. Over here, these are just the cells with the VUS showing the same thing at baseline when you activate the focal adhesions. And this is where we use the same inhibitor as we used over here to block activation of the focal adhesions. And it also prevents that cell death marker, which again tells us the direction of the arrow that activated focal adhesions are leading to increased cell death. So this is what I've shown overall. The variant, the VUS that we've been looking at, leads to aberrant aortic structure. When you add hypertension, you increase strain on the focal adhesions on the surface of the cell, and that activates focal adhesion signaling. Those adhesions are screaming out to the rest of the cell, work faster, work harder, and that increases the energy demands on the cell. And that leads to increased reactive oxygen species because the mitochondria are stressed out. And the increased reactive oxygen species leads to increased cell death, and that is what we think leads to dissection. Obviously, I haven't shown you every detail of all of the work that we did to identify this pathway, but what we're doing now and what I want to talk about is we're trying to see if all of these steps below add hypertension might be a universal mechanism for dissection, might be happening in other models of dissection. So we're trying to look at other mouse models of dissection and in human dissection tissue to see if we can show that this same pathway is activated, even if the upper part is different because there's different genetic variants or different causes in different patients. If that proves to be true, then we'll be able to hopefully come in and target treatments towards some of these steps that could help patients in the future. And with that, that's all I have. I just want to thank everybody who contributed to the work, especially science is a team sport. Everyone on this slide contributed in some way. And I especially want to thank the patients and families for participating in the research. Without your participation, we would be nowhere on understanding this disease. So this is for you.
Thank you so much. Having heard you talk about your work many times, I have never understood it so well. So that was really good for me too. I really appreciate that. And we've talked a lot about VUSs in previous webinars. No one likes them. Geneticists don't like them, patients don't like them, but seeing kind of what goes on behind the scenes, I think is really interesting. And I think we have some questions about that to get to. So I will introduce our next speaker so that we can get to her great presentation and then the Q&A at the end. But thanks so much for sharing your work with us. Okay, next, and there she is. Great. We have Dr. Jessica Wagenseil with us today, and she is professor of mechanical engineering and materials science at the McKelvey School of Engineering at Washington University in St. Louis. She studies cardiovascular mechanics, and her work is important for testing clinical interventions for elastin-related diseases and for designing better protocols for building tissue-engineered blood vessels, which all sounds fascinating. So I'm very excited for this presentation and to hear about your work, which I've heard a lot about. So I will turn over the presentation to you.
All right, thank you very much. And I will remind you all that, yes, I am an engineering professor. And so this presentation will be a little bit different than the ones that you've already seen. But I think it's also important to remember that the aorta is exposed to mechanical stresses and strains and forces. And so it has a mechanical function. And that mechanical function is determined by the materials that it's made of and how those might change with aneurysmal disease. So we use mouse models, as do the other groups, and this is an ultrasound of a mouse aorta. And what's important here is that you just saw the aortic valve open here. It's shut now. It's going to open. Oop, it just froze, of course. So we'll play it again. Well, it was open, but when it opens, it's, let me see if I can go back. There it goes again. So when it's open, the blood is flowing out of the heart and the aorta is expanding, right? And then during diastole, when the valve is closed, the aorta actually elastically recoils. And because that valve is closed, what that does is it pushes blood downstream. And so the aorta acts as this elastic recoil device that decreases the workload of the heart. And this behavior is critical for cardiac function. And what makes the aorta able to do this is what it's made of. So the materials that it's made of. And this is an image that's similar to some of the cartoons that you saw in some of the other talks. And this is a cross-section of a mouse ascending aorta. You also saw cross-sections of human aorta in the other talks. And what we've all shown you are cells, which are stained here with these blue nuclei. So layers of cells that are sandwiched in between these layers of elastin, which are shown here in red. And then you also have some collagen in green that's in the medial layer. And then also a lot of collagen out here in the adventitial layer. And so these cells make the extracellular matrix, which is the elastin and the collagen. And then it's really the extracellular matrix that's critical for the mechanical behavior. But the cells have to make it, they have to maintain it, they have to interact with it, as we've heard in the previous talks. And one of the things that we are interested in is the integrity of the extracellular matrix in aortic disease. So these are two images from a newborn mouse aorta, where now you're seeing those elastic layers in black, and then the cells and some of the other proteins are in yellow. So normal aortic wall, this is a genetic mutation that causes ECM defects. In this case, it's reduced levels of fibrillin 4. And in the newborn mouse and humans, right, this aorta is exposed to physiologic stresses. And these stresses are different than like my students being stressed out during finals time. These stresses are forces that are divided by the area over which they're applied. And so you can have stresses that kind of try to pull the vessel apart, right, in this direction that are due to the blood pressure. You can have stresses actually that act on the surface of the inside that are due to the flow of blood here in blue. And then you can imagine stresses pulling it in this direction due to tethering forces. And so the aorta is exposed to these stresses, and it must be strong enough to withstand them. And the ECM, extracellular matrix, is very important for being able to withstand those stresses. And it naturally turns over. And so cells are making this. Enzyme shown here in this little Pac-Man shape can degrade them. Macrophages or immune cells can degrade them as well. And there's a natural turnover that occurs. But in many of these aortic diseases, one of the things that happens is when there's a defect in the matrix, then you expose it to these physiologic stresses and it's more susceptible to turnover. Then what happens is there's this progressive degradation and thoracic aortic aneurysm in dissection. And so this is what happens in this mouse model, where this is what it looks like as a juvenile without the mutation. And then there's this progressive degradation otherwise. And so we're really interested in actually these two things and how we might understand the reaction to them and also possibly prevent them. So if we focus first on the physiologic stresses, the aortic wall, as an engineer, we think of it as a composite material. So if you go back to the things that are in the wall, we have those elastin layers, we have collagen fibers, and we have smooth muscle cells. And all of these things together can form a mixture in both the unloaded and the loaded state. And these things will all respond to stresses in the wall, and they may respond in different ways. So we use computational models combined with experimental results on the mouse models to try to understand how these components respond to the stresses during growth and remodeling. And so in this example, the pictures on the right with the dots are experimental results, and the pictures on the left with the lines are some model predictions. And so we do experiments where we take a mouse aorta and we measure the pressure versus diameter. So the black line is the wild type mouse or the normal mouse. This light gray line up here is a mouse model with an aneurysm. So you can see it's got a very large diameter compared to the normal. And then this dark gray line here is the same mouse that's been treated with a drug called losartan that has been of interest for treating Marfan syndrome. And so it reduces the aneurysm, it's smaller, but the dark gray and the black curves are still not the same. And if you convert this data into what we call stretch versus stress, then this type of curve actually tells you more about the material. So in the upper curve, you can imagine a thicker tube or a bigger tube, right, is going to have different behavior up here. But down here, if the tube is made of the same material, it will look similar in this curve. So if we go back to the control, the black curve's here, but in our animal model, even when we've treated it and reduced the aneurysm, it still has very different behavior down here. And so we then try to understand with modeling what could be happening. And what we find is that we can reproduce these results of this animal model, either in the untreated or in the losartan treated state, with an alteration in the material constants of the elastin and the collagen. So basically the way that those materials are contributing or their properties have changed due to the mutation. But then it's actually only the untreated one that we can model this dilation, but the similar properties, if we have a change in the response to the shear stresses. And so the shear stresses are from the flow of blood. And so what we're predicting here is that the cells in that wall, due to this changed material, now respond differently to their mechanical environment. And then things like Callie was showing, there may be downstream signaling that then causes this dilation. And so we're interested in separating out some of these things so that we can perhaps go look at mechanisms. And the way we plot those previous graphs, we assume the aorta is a cylinder, which we all know that it's not. And that first movie showed you that it's not. It's very curved, it has branches. And so we use some more sophisticated modeling that are called fluid-solid interaction models, where we take MRI of the entire mouse aorta. We can then isolate the entire thing to look at the geometry. We take out pieces of it to do look at the dimensions and to do the mechanical properties. And then we can model all kinds of things, where in this case, we're looking at the von Mises stress, which is a measure of a maximum stress. And so you can see where the maximum stresses are on this wall, and that might be indicative of that where there's going to be a problem, and when it reaches some maximum amount that we might want to do an intervention. And so we've used these types of models to look, again, in our mouse models, to look at a variety of what were called mechanical biomarkers. In this case, it's oscillatory shear stress. And so you can see the red means that it's high, and you can see some aneurysms are big, some are small. And so we're trying to understand how these shear stresses relate to the aneurysm growth and actually to the outcomes. And in this study, we used lifespan over here, plotted on the y-axis, as a version of outcomes. And we were trying to understand if any of these biomarkers can predict lifespan, right? So that if you could measure, in this case, this is time averaged wall shear stress or oscillatory shear stress, if you could measure that using imaging, which you can do in humans, could you predict outcomes and try to design an intervention? And in this case, these two mechanical biomarkers correlated better with lifespan than one of the clinically used parameters, which is simply maximum diameter. And the other thing we found in these studies, if you remember these two curves that I showed you here, where we had the drug was reducing the diameter but not changing these stretch versus stress curves, is because it wasn't changing those material properties. And part of that, this is the group over here that was treated with losartan, and this is the untreated. So untreated has really disorganized elastic fibers, but even the treated ones, the arrows are showing these breaks in these elastic fibers. And so we still haven't fully recovered back to that material of what we would really like the aortic wall to look like. And so if we focus very briefly on ECM turnover, and the idea that when the ECM has a defect, it's more susceptible to turnover. And we are trying to understand if we can try to stabilize this turnover and prevent moving from this picture over to this picture. And one of the things that we have looked at is a drug called PGG, or pentagalloglucose. And it has been used starting 15 years ago to stabilize abdominal aortic aneurysms in animal models, where this is a chemical model that creates an aneurysm at day 0. If you put saline on the aneurysm, up here it grows 40%, but if you put PGG on it, it doesn't. And down here in the PGG case, we have maintained this elastic fiber integrity that is not there in the saline case. But abdominal aortic aneurysms are a very different disease than many thoracic aortic aneurysms and dissections. And there's a lot going on in vivo. And so we wanted to go look at just elastic fiber stabilization in an in vitro model. So we did our mechanical tests on the mouse arteries, the aorta. We use a drug, elastase, that degrades the elastic fibers. We also tried just the PGG alone. And then we try to use the PGG either after degradation or before to see if we can alter the mechanical properties of the aorta just by addressing these elastic fibers. And the upshot is that the gray curve is our pressure diameter of what a normal mouse aorta looks like. If you just use the PGG, it doesn't change anything. When we use this elastase, you can see this huge dilation, and then it becomes very stiff and large as we increase the pressure. And the PGG used either before or after brings the data back to some intermediate point. So it can partially help these changes in mechanics, but not completely. And if you go look at the wall structure, here's the untreated with our elastic fibers again, and collagen out here. And our elastase treatment that's degrading the fibers causes these breaks, and then also causes this buckling or kind of disconnection between the elastin and the collagen. And the PGG treatment is partially restoring both of those. And so we think that that kind of larger treatments that address some of these structural defects might be another way of addressing some of the downstream signaling that is affecting the disease. So we use mouse aorta, and we look at mouse models that have incorrectly formed or cross-linked elastic fibers. And they have all of these changes in mechanics, basically, that may lead to signaling cascades that contribute to disease. We use mechanical analyses and modeling of varying sophistication, so from a cylinder model all the way up to complicated fluid-solid interactions, to look at disease biomarkers and remodeling stimuli. And we think that stabilizing some of the elastic fiber degradation or even other ECM defects may be an upstream target that prevents some of these downstream effects. And as was mentioned earlier, I do not do this in a vacuum. I have many lab members, current ones who are shown here in this picture, many collaborators, and many funding sources that have helped with this work. Thank you.
Fantastic. Thank you so much for sharing your work with us. Really interesting. I haven't really seen too much of this side of the engineering side, so that was really interesting for me. If the other panelists want to jump back on screen, I can ask y'all some questions now. And we have a few in here. So the first thing I want to ask, because a few people did ask this, but this is for all of you. So you all work on mouse models of disease. And someone asked a good question, which is, can you translate that directly into a human? Obviously, we are not mice. Presumably, there's important biological and mechanical differences between how a mouse aorta works and a human. How do you take what you learn in a mouse and move it to human treatment or care, which of course would be our goal in the end? And sorry, by not singling anyone out, I've made everyone hesitate to ask. So maybe I don't know if anyone wants to raise their hand and volunteer to go first. Okay, Alex, go for it.
One of the beauties of working in a cardiac surgery lab is that we have access to a lot of tissue straight out of the operating room. So when Dr. Fishbein does some of these aortic aneurysm repairs, we'll be able to get tissue, whether it be different connective tissue mutations or even just patients who have had type A dissections. We have access to that tissue. And so we're trying to integrate a lot of what we've learned in the mouse models, which is very controlled, and translate it into what we're finding in the human patients. And so that's an ongoing process, but having that access has really enabled us to make that jump into more translational medicine.
I don't know if either of the two you want to comment also.
Sure, I can say from the discovery side, while we're trying to figure out what's going on, like Alex said, the most important resource is just also using human tissue and cells to try to validate what we're finding in the mouse and making sure that we see the same things regardless of species. But in terms of trying to develop treatments, often there are steps in between where you will try a larger animal model before going directly from mouse to human, because there are a lot of differences in terms of how the aorta functions between the two.
Okay. Brian has asked, therapies for those with aortic aneurysm all seem to focus on reducing stress on the aorta caused by blood pressure and force. Are there therapies that can assist strengthening the aorta? I know and also there are some surgical procedures where the Pears procedure, which Alex is probably familiar with, where you coat it, that some people have talked about in the past. Can we make our aorta stronger?
Yeah, I mean, so I'll let Alex chime in from the clinical point of view, but from the mechanics point of view, right? Yes, I mean, so one of the things I talked about was trying to reduce this degradation, which is one way of making it stronger. There are certainly other people focused on, can we make more functional elastin? I think, one of the problems is there's fibrosis is actually going the opposite direction, right? So if you lay down too much collagen on too much matrix, it can become really stiff, which is another problem. And so there's a very fine balance between where it actually needs to be for the proper function. But yes, it's an active area of research. And in terms of diet and exercise, that's good for you overall. I don't know. I'm honestly not familiar with the evidence on, how it certainly strengthens the heart. I don't know how about the aorta too much.
If people do want to, or perhaps one of our folks in the chat can add in, we did a webinar with Dr. Murdoch recently, and he talked about things, and so did Dr. Prakash. So maybe I can point to that are exercise specific, because they are working in that area. So I'll add that to the chat if people have further questions. Did you want to comment also, Alex, or we're good?
Yeah, from the clinical side of things, for the most part, as surgeons here at Stanford, we're replacing the aorta. So that's our major toolkit, is when someone has an aneurysm, to prevent rupture or dissection, we prophylactically replace. And that's why we're spending a lot of time in the lab trying to figure out how we can lengthen the time of when we diagnose an aneurysm and when we have to replace, to even preventing the aneurysm from forming in the first place. But I think a lot of work still has to be done to get there. So right now, we really only have the one very invasive tool from a surgical standpoint.
Mm-hmm. Callie, as I was watching your presentation, and I've heard you talk about this over your work over the years, since I used to work in the same lab as you, I wonder though, a question we get asked a lot is, how long is this going to take? How, and watching your work, can you give an estimate of how many years of work you presented in the 15 minutes? How long did that take you?
The project that I presented to you guys today actually was the end of my dissertation research, and I defended my dissertation in 2013. So it's 10 years of work, essentially.
So when people rightly, are feeling anxious, they want an answer, they want to figure out, especially people with VUSs, I know it's a really hard place to be in that. What is the meaning of that? And behind the scenes, though, to know that people are spending a lot of time and effort to figure out answers to these questions. Okay.
We also wish science would move faster, believe me.
Yes, you all also want it to move faster. Great. Okay. Given insights on stretch, so I'm guessing this is for our engineer. Is there any known or anticipated relationship between having varicose veins and risk for aortic dissection or aneurysms? I'm not sure if you've ever done work on.
I do not know. Yes.
Okay, I don't know either. That's an interesting question. I don't know who asked that, but if you want to email it to me, I might be able to get it to someone who works on varicose veins, and we can get you an answer. Okay. Are single cell studies only for people who know their gene?
No. So the beauty of what we're doing is it's kind of like a shotgun approach. We're taking any tissue that we can find, chopping it up into single cells, and running it through our system, and sequencing the RNA. And so it will tell us what genes each cell has. And so if you have a cell, we can sequence it essentially. And we're looking at Loeys-Dietz, Marfan. We haven't quite gone into any of the non-genetic aortopathies yet. But that's something that we hope to sequence soon.
And I actually had someone email me during the presentation and said they are having a planned aortic repair, sounds like fairly soon. They wanted to send us, which we cannot accept your aorta. I'm in my own home, so please don't send it to me. But I guess a general question is, if you are a patient who is going to have your aorta replaced, can you send it to Stanford or UT Health, or I assume there's someone at WashU who's also accepting aortas? Is there a way as a patient you can proactively say, take my aorta?
I think there's a lot of sort of regulations to jump through to be able to send tissue in between institutions. So I don't think that's quite possible yet. But hopefully in the future, if this technology gets more integrated into the clinical side of medicine, maybe one day that will be useful for a diagnostic tool.
I will caveat that and say, yes, on the timescale of doing single cell studies, that may be true, but we have gotten tissue from patients around the country preserved for histology or frozen for RNA and protein studies. And so if it's something that you're interested in, the first step is to enroll in the research study of your choice. regardless of whether know your genetic variant, have family history, anything like that. There's probably a study out there that would work for you, and enroll in the study and then offer whatever you are willing to offer in terms of your participation. And that definitely helps move the science forward.
And I know that I think Bella Morin is on this chat, who is a research nurse for the Geometer Research Program, which you all probably know about if you've been playing along with us on these webinars. So that program does accept, if it can work out, samples and whatnot. I see we are at the hour here, so I'm going to, unfortunately, we still have some questions, but I respect your all's time, and I know you have important work to do since we saw all of it, and we want you to get back to it. Thank you so much to all the panelists today. I see people in the chat are saying thank you for your all hard work and that it was very informative. So I think people really appreciated seeing kind of behind the scenes what is happening that sometimes patients and families aren't too familiar with. So thank you for being here. And this, as I said, is recorded, so it will be available to view later. Someone did ask if they would be able to look at the slides. I don't know if y'all are comfortable sharing your slides with me or not, but if so, I will also post those on our website. And yeah, Bella's in the chats with her email if anyone has questions about research. So thank you again. We are going to actually be back May 10th with another webinar. This will be more clinically focused. We're going to talk about genetic testing in the postmortem setting. So if you lose someone to dissection, how can you make sure that genetic testing and evaluation is done for the family? So changing gears a little bit, but I hope to be back to this side of things in the future. Thank you all again.
Thank you.
Thank you.
Thank you.